All right, ladies and gentlemen, I am Sarthak Kaul, and this is True Calling. Our guest today is Dr. Satish Pillai. Dr. Satish has spent a few decades uncovering one main question. Despite years of research done in the field of HIV, why is there no cure for it? Satish is a leading researcher in HIV cure research and global molecular surveillance of blood-borne infections at UCSF and a senior investigator at Vitalant Research Institute. He was an associate investigator of the Blood System Research Institute, also called as BSRI, that is the second largest blood banking company in the US. BSRI has helped develop a number of viral infectious disease screening tests, including ones for hepatitis C, hepatitis B, Zika, West Nile, and HIV. Along with that, Satish is also an avid musician with a deep family history in music. So, in our conversation, we discussed his family's background in Carnatic music and mathematics, his journey moving from an early interest in astronomy to biology, why it's so hard to treat HIV, and the topic around viruses, problems with citations, social media, and much more. I hope you guys enjoyed this conversation as much as I did, and I'll see you guys in the next one. When you look at yourself, how do you introduce yourself? What do you say? Who is Satish?
I guess I would define myself as a scientist and a musician, and both of those things factor pretty equally in my life. So that's how I would a scientist and a musician, and I'm a dad. Those are my favorite things to represent.
That's super interesting. You said something right now, which was like people are wearing AirPods, and I was having this conversation with someone. He was like, I want to tune in to kind of be locked into whatever I'm listening to whenever I'm doing whatever. But for me, it's the perspective that you have, which is like I don't think it's as much tuning in for most people as it's tuning out.
I completely agree with you, and I think randomness is such a rich experience in life, just randomly bumping into people, frankly eavesdropping, listening to other people's conversations when you're sitting in a coffee shop. I love doing that, just hearing what other people are gabbing about. And when I just kind of look back through my life, I think most of the best things that have happened to me have been through just random chaotic interactions with strangers. And I think that when you wall yourself in with, wearing headphones or AirPods all the time, you just don't allow that to happen. And I think that's a shame. And yeah, I think it's at the heart of a lot of our social problems because we relate to each other less and less because we're not just open to randomly entering into what are often very meaningful conversations. And frankly, even the meaningless conversations are meaningful.
Yeah. I mean, I think serendipity has such a huge part to play. Have you had a serendipitous interaction this year?
Oh, this year. Okay, I think you're saying general. So this year, that's a good question. Is there a particular serendipitous interaction that well, actually, one that I would describe as serendipitous would be with Aristotle, which is, how ultimately I find my way to you. And actually, that, if I could rewind the tape a little bit, the way that I know Aristotle is one of my best friends in the world is this guy named Josh. I play in a band with him called Boy in the Bubble. And the way that we met, probably 14 years ago or something, is that I used to perform at this coffee shop in San Francisco, and he used to perform at the same coffee shop, but we'd perform on different nights. And the woman who ran it was like, hey, I feel like you'd really hit it off with this Josh guy. And I was like, sure, okay. So, we ended up hanging out, and then I ended up, joining his band when we've been playing in a band together for ages. And it turns out that Josh went to Columbia with Aristotle and thought that me and him would have some really interesting scientific interactions. And so that's how I met him. And then actually it's been really wonderful meeting him and learning about his whole world with in the private sector, which is very different from my academic space. And that's how I wound up here talking to you. So that I think is a great example of a kind of a serendipitous chain reaction there, right?
Man, that's awesome. So what are the kinds of conversations you have with Aristotle on the science front?
Well, so I think really what they're focused on is on the future of science, and the future of how science will be conducted, and the role that more and more, which is obvious. The intersection between science and computation is huge, right? So what I think is a huge problem in the way that science is practiced now is everybody has the capacity to generate massive amounts of data. my lab, everybody lab. There's thousands of labs around the world that are generating tons and tons of data, but the bottleneck is in the analysis and the intelligent interpretation of those data, because there's not enough analytical expertise to deal with all of the data that are being generated. And so most of my conversations with Aristotle have focused on that in terms of how computer science and the field of informatics can engage scientists, like life scientists, for instance, in a more productive manner so that we can get rid of that bottleneck. Because that would change. So just to give you an idea, in terms of how we could develop new therapeutics for like emerging pathogens, all that stuff, a huge roadblock is the data analysis capacity that we have in science. And so if we could essentially push the turbo boost button on the data analysis, it would dramatically increase the rate at which we could develop new therapeutics, vaccines, that kind of stuff.
So let's get into that. I'm very curious to, understand more of your research. I guess the place to start is, what does someone need to know about your story to understand why you do what you do when it comes to the research you're doing?
So I'll start way back with my parents. So my mom, who's definitely, one of my all-time heroes. My mom was a mathematician. She's not actively doing math anymore, but when she was a teenager in India, she was working for the Indian equivalent of NASA, which is NAL. Are you familiar with NAL, the National Aeronautics Laboratory? Yeah. She was using math to do stuff like calculate rocket trajectories and stuff as a teenager. And when she was super young, I think 20 years old, she was recruited by pretty much every highfalutin graduate program in the U.S. to do a PhD in math. And she ended up coming to Brown University at the age of 20 to pursue her PhD in math. And that's where she met my dad. So my dad came over to do a PhD in biochemistry at Brown. And so, they met over there. So both of my parents, with both me and my sister, they were always steeping us in math and science. That was a big part of my environment growing up. My mom is also a fantastic vocalist, a Carnatic vocalist. And so she set up this magical music culture at home, which is why I think both me and my sister are super into science and super into music. And so they went out of their way to expose us to astronomy, math, everything, life sciences. And so that's the main reason why I think I wound up being a scientist, is a lot of inertia from being a child. My dad had a clinical laboratory where they were testing blood, like doing standard blood work and also testing for bloodborne pathogens, that kind of stuff. But he brought home like a Zeiss microscope when I was a little kid, and so from a young age I could just walk around with slides and cover slips and grab little pieces of stuff, put it on a slide, and then look at it under the microscope. So I think that's how I really got into science as a kid. And in terms of what I do now, I'm a virologist, and kind of how I got there, I'll give you the cliff notes. As an undergraduate, so I went to the University of Arizona to study astronomy. And I was listening to your podcast and kind of like your evolution through physics, and there's some commonalities there. I think I had a pretty stupid, immature concept of what studying astronomy was going to be like. Like I think that part of me thought I would show up in Arizona and they'd be like, Here's the key to the observatory, and a bottle of wine. Go up there, son, and have some fun. And of course, it's not like that. You're basically you're doing math the whole time. And at the end of the day, my mom is a math genius. I have other math geniuses in my family. I'm not a math genius. I'm okay, but I don't think I'm a terribly gifted mathematician. And so I don't think I was entirely satisfied with doing astronomy. Serendipity. So to bring it back to serendipity, I took an environmental biology class 2 years into college that was taught by this wonderful professor named Bill Calder. And this dude just practiced what he preached. Like, he was a hardcore environmentalist. He built this home that was completely green back before it was hip to have a greenhouse. He was off the grid. Everything was run off of gray water, all the stuff. He would take us into the mountains, like in the Catalina Mountains north of Tucson. And, we would do these awesome like ecological surveys up there. But he would do this thing that was so magical. He would stand at like a bottom of a tree and he would do bird calls. And he would literally bring several different species of birds to the tree with these bird. He literally just sounded like a bird. Like, you wouldn't be able to tell he wasn't a bird of these different species. It was nuts. He just was so inspirational that a couple of years in, I switched from astronomy to biology. And I specifically switched to what's called ecology and evolutionary biology. So that was my major, and I totally loved it. I love studying evolutionary biology. So I was 100% locked into the evolution thing. And so I did a lot of undergraduate research studying the interaction, the coevolutionary relationships between plants and insects. So just to explain that kind of succinctly, there's this constant evolutionary arms race. There's essentially a conflict between plants and insects, right? Because insects often lay their eggs on plants and then their larvae hatch and then they eat the plants, right? And so it's a huge problem for agriculture. So there, agricultural pests that just decimate crops. And so I was working specifically on this insect called the diamondback moth. And so it would lay its larvae on specific plants, and then their larvae would just chew the plants up. Game over, right? So there's this constant evolution where plants would develop, they would change kind of the chemistry of the waxy cuticle of their leaves to repel the insects, but then the insects would evolve to kind of deal with this new repulsive waxy coating, and there'd be this interesting coevolution that would happen between plants and then what's called their phytophagous insect hosts. So I got super into that. And then towards the end of college, I started reading about host and virus coevolution. And the reason why I got so interested in that is that most of the things that you study in evolution, let's say you're studying the evolution of marine mammals or something, most of these phenomena occur on timescales that are basically irrelevant to the human mind, right? They're occurring over millions and millions of years, and you can't really watch anything happening in real time, right? Most of it is very speculative. And the thing that really attracted me to viruses is that you can just watch viruses evolve in real time, like so quickly, right? Either, in the natural world in terms of how viruses become resistant to drug therapies or become resistant to vaccines, or in a petri dish, right? You can grow a virus in a petri dish and challenge it with something, and you can just watch it evolve so quickly. And so I got really, really interested in this idea of studying the evolution of viruses and their hosts. And again, serendipity, I was finishing college, and then I'm just old enough where, like, I think email was basically like invented, and it wasn't invented, but it was like it became a thing for like normal humans to use when I was finished. I don't know if you remember what Pine is. It was like it was a text-based, wait, was it Pine? So the original like text kind of interface was called Elm, and Pine it stood for Pine is not Elm. How nerdy is that? But I remember using on this Pine email server, this message came from Los Alamos National Laboratory, and it was from an amazing scientist named Dr. Betty Korber, and she was advertising for a position in the theoretical biology and biophysics program at Los Alamos, and they were looking for graduate research assistants who are interested on doing computational work on viruses. And I applied, and then I got very lucky. I did a phone interview. They flew me out for a in-person interview. Side rant that has nothing to do with the story, but I had like one pair of decent pants, and in the morning I spilled a whole cup of coffee on myself, and I couldn't wear those pants because they were doused in coffee, and I only had one other pair of pants, which was a pair of bright green jeans that were like cut off. Ridiculous. I was a total idiot. And so I showed up with this very nice dress shirt and I think even a tie or something tucked into this pair of bright green jeans, thinking, I'm screwed. I'm not going to get this gig. And then I showed up there, and then I asked the person at the front desk, could you please tell everybody to just pretend that I'm a torso for the rest of the day? And they're very nice. I think those green pants actually helped me get hired, to be honest.
Is that why your favorite color is green?
It is. Green is definitely, I think, my favorite color. Yeah, it really is. I love the color green. And I had these just obnoxious bright green jeans that no sane human would ever wear to a job interview. But that's what I wore, and they took pity on me. But so I got this job, and it was doing computational work, 100% dry lab. So there was no wet lab there. We were just doing computational work on mainly HIV evolution. So the evolution of HIV and other related retroviruses. And it was awesome. So I spent a couple of years at Los Alamos National Lab training there, and that just kind of set the stage for what I did. So ever since then, I've been doing kind of a 50-50 mix of like lab experimental biology, specifically focused on viruses, and then also kind of bioinformatics and computational work on viruses, and mainly focusing on HIV, with some parsley on the side of the plate, but mainly I've been focusing on HIV/AIDS.
Thank you for that breakdown. I mean, that gives so much more light to this. So,
yeah,
there's, you've said this before, despite 3 to 4 decades of focused research since we discovered HIV-1, there's no cure for it or no vaccine. So what is unique about HIV?
Yeah. So a lot of it has to do with what is unique about retroviruses, which are the family that HIV belongs to. So HIV does one very nefarious thing that's part of the life cycle of all retroviruses that makes it very, very difficult to get rid of. So when HIV infects one of your cells, it actually integrates its DNA into the genetic backbone of your cell. So it kind of becomes part of the genetic identity of your cell. And so it is very, very, very difficult to get rid of something that's been integrated into your genetic information. That's one of the reasons why it's very difficult to get rid of. The other thing is once it integrates itself into your genetic backbone, a lot of times the virus can go into a relatively dormant state. We call it viral latency. The term viral latency I kind of almost hate using because the term latency has many different meanings, actually. But here, what we really mean is that the virus can enter some sort of quasi-dormant, quasi-stable equilibrium where it's not producing much of anything. And so what happens there is your immune system doesn't even recognize that cell as being infected. So, once your immune system can see an infected cell, it'll do something to target and destroy it.
Like, imagine I'm like a 10-year-old kid. I have no idea. I actually have no idea about how this works. So what is, how does our body detect something that's not within our body and it's new?
Excellent, awesome question. So that's kind of the whole gig with the immune system, right? The whole gig with the immune system, um, which, by the way, is obviously not unique to humans. So there are two kind of gross branches of the immune system. One is called the innate immune system, and this is what is kind of thought of as being the primitive, sort of primordial immune system, and it makes a very crude distinction. The distinction is self versus non-self. And so basically, that's the only distinction that can be made there is when you have an invader, it can see something as being not you, and then it destroys it. Then we have this thing called the adaptive immune system, which is way, 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 way more complicated and nuanced. There, your immune system, rather than making this crude distinction between self and non-self, it's actually designed to recognize very specific sequences, very specific, unique sequences. So there you'll have an immune cell that's specific to one strip of amino acids from influenza or one strip of amino acids from SARS-CoV-2. So that's like a much more subtle, targeted, precise assault on specific pathogens, right? Those are kind of the two ways that the immune system targets and destroys things. The way that most of this works is through really pattern recognition. And I'm guessing like through your background with physics, you must have delved into computer science, and there's a lot of pattern recognition there, right? So really the way most of the immune system works is through recognizing patterns that are the host and being educated in a manner where you don't react to it, and recognizing patterns that are associated with an invader, and then using that to destroy something. And so I'll give you an example, like with what I was calling the adaptive immune system, where we have all of these immune cells that target very specific sequences. If we look at T cells and how T cells are generated, what T cells are one of the main type of immune cell. They're educated in the organ that we have called the thymus. And what happens is if you have a T cell that reacts too aggressively to one of these self proteins, it's eliminated from the pool. Because if you let that T cell leave the thymus and then populate your immune system, then you develop autoimmune diseases, right? Then your body would start attacking yourself, and then you're in big trouble. And so the immune system has all these methods to educate the cells that are in development to make sure that they're reactive, but not reactive to bits and pieces of yourself so that you don't attack yourself. But obviously these things go wrong because we do have lots of autoimmune diseases, right? And so
so what happens there?
What happens when you have autoimmune diseases? So when you have autoimmune diseases, one or more branches of your immune system fails to make a proper distinction between what is you and what is not you, and it starts attacking your own tissues. So my wife is actually, she'd be much better talking about this because my wife is a rheumatologist and an immunologist. She's a rheumatologist who treats people with autoimmune disorders. And so, most of her patients have like rheumatoid arthritis or lupus. Those are kind of the most common conditions. But that's really where your immune system, goes off the rails and cannot make a proper distinction between self and non-self. So the question is, how does that happen? We have all of these safeguards in place that prevent that, right? So here's one theory, and I think I would still say that it's a theory. I don't think it's like a proven fact, but I love this theory because it kind of brings it back to my virology universe. But one of the theories with specific autoimmune disorders is that we get exposed to some virus, let's say. We get exposed, we get infected by some virus, and one of the viral proteins looks very similar to one of your own host proteins. And because your immune system, builds a robust response against this foreign invader that has a protein that happens to look like one of your own proteins, then your immune system starts attacking your own self, because there's kind of like a mimicry between the viral protein and the host protein. So there's a whole theory that probably not all autoimmune disorders, but many of these autoimmune disorders could be caused by viral infections that initiate an immune response that ends up turning towards a host, which pretty crazy concept, right? And actually there's some theories even that, like diabetes, for instance, that diabetes could be triggered to some extent by a viral infection where the virus has some protein sequence that's similar to what you find on a beta cell that produces insulin. So your immune system starts attacking and destroying your beta cells, and so you lose your capacity to produce insulin. And so really, we're always walking on this precipice, I think, evolutionarily, of having a robust enough immune system to fight off this soup of pathogens that are trying to make a living, in us. And again, if we tip the scales and our immune system is too robust, then we start destroying ourselves. So the immune system has to be finely tuned to be vigilant enough, but not too vigilant. And there's so many examples in nature of how, these things, there's a trade-off between these things. So like, for instance, there's specific immune genes, like variants of genes in our immune system that are associated with really great viral control that also happen to be associated with autoimmunity. And so I'll give you one example. So we have these genes in our immune system called HLA genes, like human leukocyte antigen genes. And what these genes do is they which protein sequences we're capable of targeting and destroying. So they kind of fine tune which protein sequences we're going to make a robust response against. And there's one variant of an HLA gene called B57. This B57 variation of this gene is associated with excellent control of certain viruses. So people that have this gene tend to have lower viral loads in the setting of HIV infection. So meaning that they have lower amounts of the virus because their immune system is better at controlling the virus. And then they progress more slowly to severe disease when they have this gene. But that HLA-B57 gene variant is disproportionately represented in people that have rheumatoid arthritis and other autoimmune conditions. So it's a perfect illustration of how a gene that's well adapted to control foreign invaders, control these viral pathogens, actually is maladapted when it comes to autoimmunity.
Very interesting. So on this topic, first question, and I'm asking all the basic questions here. It seems like the virus has its own intelligence. Like there's a level of intelligence to it. What are your thoughts on that?
Yeah, so it's hard not to think that, and I have to say, like, as somebody who's been studying HIV evolution now for decades, okay, this virus has a tiny little genome. Okay, it's got its genome is less than 10,000 base pairs long, and it has like 9 genes, right? Wow. And it effortlessly hijacks a human being, that has 30 to 40,000 genes and billions of base pairs in its genome, right? So it uses so little to hijack such a complicated thing. And really what you can see is no matter what the immune system throws at it, the virus just figures out a way to slither through it by mutating and also recombining. So that's another magic way that HIV and other retroviruses can evolve. In addition to just developing mutations, the virus can kind of shuffle its deck by taking bits and pieces of different viral genomes and forming novel mosaics, right? And so all the time I personify the virus and I think of it as like this evil genius. But the truth is it's kind of exactly the opposite. It's actually the ultimate, the phrase throwing spaghetti at the wall. That's what RNA viruses like HIV do better than anything, is they just throw tons and tons of spaghetti at the wall. Meaning when HIV makes a copy of itself, part of its programming is to make mistakes. So every Xerox copy of the virus that's produced has some mistakes in it, has some mutations in it. And really the way that HIV survives is just by making tons and tons and tons and tons of mistakes And by chance, one or two of these variants that have these mistakes in it can survive whatever you throw at it. So it's kind of the opposite
Wow.
of intelligence on some level, right? It's just you
Randomness.
it's just riding this awesome randomness wave where it just creates lots of noise. And so you're just constantly generating this spectrum of variants. And just by sheer chance, one or more of this kind of probabilistic cloud that's generated is going to be able to survive whatever onslaught comes down the pike. So in some ways, it's kind of the opposite of
Whoa.
intelligence. It's like a brute force method, right?
Yeah. Yeah. I mean, it's just that's why it's so hard because of its random nature.
So it's really evolutionary wizardry, so it
Yeah.
can evolve so quickly to circumvent whatever you throw at it. That's one thing. The second thing is the fact that it becomes integrated into your genome and becomes hidden from the immune system. I think those are kind of the two biggest challenges. And because the virus evolves very quickly, one of the consequences of that is if you look at the global genetic diversity of the virus, just kind of all of the different ice cream flavors of the virus comes in,
Hmm.
there's a massive amount of variability there. So that creates a huge challenge for developing like a preventative prophylactic vaccine, right? So when you try to develop, vaccine antigens, it's pretty difficult to come up with something that's going to represent all of that worldwide diversity that's out there. And so, to give you an example, the flu vaccine, right? We have our seasonal flu vaccine. If you look at any 5-year period, probably one out of those 5 years, despite the best efforts of the people out there who are designing the flu antigens, they can't accurately predict which strain is going to blossom and become the dominant strain of that season. Probably 10-20% of the time, the flu vaccine that's distributed doesn't really line up very well with what becomes the circulating dominant flu genotype. And so there's not that much cross protection, right? And that's with a virus that's not nearly as variable as
Hmm.
HIV. Like, HIV is several times more variable. And so the challenge there is monstrous. But actually, there's one other thing. So, I mentioned the integration into the host genome. I mentioned the kind of hiding from the immune system, the
Hmm.
genetic diversity. The other thing, which is somewhat surprising and frustrating more than 4 decades into this epidemic, is that we don't really have an excellent understanding of what a good protective immune response is against
Hmm.
HIV that keeps you from getting infected. That's the truth. The language again is correlates of protection. We always talk about that in the infectious disease space. If we want to develop a vaccination strategy to prevent somebody from getting infected, what we need to identify is a correlate of protection, meaning that we need to find people that are naturally not getting infected in the absence of vaccination and figure out
Hmm.
what their immune system is doing to allow them to not get infected. And then we need to study them deeply to figure that out. And so I'm sure you've heard this tons of times. I'm sure you've heard tons of cases where there's been a household where way before we had vaccines for COVID, everybody gets infected except for one person in the house is peachy. Everybody else is sick and coughing up a lung, and one person just manages to be peachy. Well, there's probably something immunologically interesting about that person, that they had some immunologic gift that prevented them from being susceptible to the SARS-CoV-2 infection, right? So that's kind of what we need to do to develop prevention strategies, is we need to identify people that are naturally resistant, figuring out what that magic sauce is and figure out how to replicate that in other people. And I think with HIV, to this day, there are people that are very actively focused on this, but we don't have a clear idea of what those correlates of protection are. So we don't know exactly what to mimic in a vaccine. And a lot of it is because it's very difficult to identify large numbers of individuals that are continually exposed to HIV, but they don't actually ultimately acquire the infection, which is not a trivial thing to do.
Are there any demographics, any populations that we already know of that have had some success with this? In the sense, like they just like have a proclivity to not acquire or get affected by HIV to some level.
Yes. So there's one glowing example of this. So, and I'll explain to you kind of what the biology is first. So, if typically infects that immune cell is talking about T cells, right? So T cells are kind of these, they're kind of like the cavalry of the immune system. They're one of the most
Hmm.
important soldiers in the immune system, right? So HIV actually makes a living in T cells primarily, not exclusively, but that's kind of the main cell that HIV infects. And so one question is, how does HIV enter these T cells, right?
Hmm.
So it turns out it depends on a couple of different proteins. So it kind of uses them as a door to enter the cells. And one of those is called CCR5. And what CCR5 does is it plays an important role in what's called immune cell trafficking. So let's say you get infected with some bacteria, right? You get a bacterial infection at a specific site. So it turns out CCR5 plays a very important role in responding to chemical messengers in your body so that they can traffic to sites where they're needed. There's a natural occurring mutation called the CCR5 delta 32 mutation. And so what happens is when you have that gene does not encode a functional version of that protein. So the protein is misfolded, it gets immediately degraded, and it never shows up on the surface of the cell. But it's more complicated than that because humans are diploid, right? So we have two different versions of this gene because we have two copies of our genome. Some people will have one messed up version of this gene, so one CCR5 delta 32 allele, and then they'll have a normal allele. So they'll have a matching CCR5 delta 32 gene version that works. And so those people are, I can't say 100%, but they're like approaching 100% protected from HIV infection. They almost can't be infected by HIV. So once in a blue moon, individuals that are homozygous for that CCR5 delta 32 mutation still do get infected, but it's very rare.
Are there any patterns there in terms of what these people, like, is it just evolutionary? Like, is there anything happening on the nurturing side which is like leading to this?
So I guess one question is like, why do these people have this mutation?
Yeah.
I mean, so that's a critical question because obviously, HIV is just a little blip in human evolutionary history. According to the mathematical modeling, HIV has probably been, it probably entered the human population about 95 years ago. That's totally insignificant in terms of human evolutionary history. So the theory that was presented, maybe somewhere between 10 and 20 years ago, that was a super cool theory, which has been kind of partially debunked. So one question is, where do you find the CCR5 delta 32 mutation? It's not evenly distributed across humans. So you find the free CCR5 delta 32 mutation mainly in northern latitudes in Caucasian populations. That's where you find it primarily. That's where it's at high frequencies. And so it clearly has something to do with the human evolutionary history in that region, or at least in people with that evolutionary background. And so one of the theories that was put out a long time ago, which I think is a cool just so story, but I don't think it's ever been proven, is that the CCR5 delta 32 mutation conferred resistance against the plague, against the Black Death, right? Against the plague, which obviously, the plague decimated humanity, right? And so the theory is that if you actually look, and a lot of this, there's a lot of circumstantial evidence that kind of cements this theory that if you look at the geographic overlay of where the Black Death was really prominent and the CCR5 delta 32 mutation, you do see that there's a good kind of overlap. There's a lot of circumstantial evidence that suggests there's a possibility, but it's certainly never been proven, and it's very controversial. The long story short is we don't really know why the CCR5 delta 32 mutation exists in the human population, but there's a very good possibility that it has to do with ancestral conflicts with other pathogens. And I can tell you one thing that's super cool is that in general, the genetics, human genetics that are associated with the immune system that kind of determine what the nature of our interactions are with pathogens in the contemporaneous world. And there's lots of cool evidence of this.
So I have a few questions here. And these are questions that I think might be too dumb. Before that, I read that when you were actually pursuing your grad school, you at one point were going to drop out.
Multiple times.
Yeah. So what was happening in that time for you, first of all? What was going on in your mind? But also, what changed for you to continue and pursue that? The reason I ask you this is there's too many people I've met that are in their 20s that my close friends are also pursuing their PhD, and they've had this thought like, hey, I don't know what the hell I'm doing here. I don't understand where to go from here. What is something you can tell them about from your journey?
Yeah, and I think from listening to kind of your introduction that you have on your YouTube channel, we probably had some similar feelings of disillusionment during our careers. So, okay, there's a few different components of this. So one is like, what is my motivation for doing this in the first place, like doing a PhD in the sciences? A lot of it was definitely like organic interest in the subject matter and a passion for it. There's no doubt. But then some of it, no doubt, is my culture as a second generation Indian kid, like there's so much pressure as part of it, but there's also like there's a concept that, either going to medical school, which clearly was not my gig. So I never had any, fallacies about that. But that being a doctor or being a scientist, that these are like respectable professions. And so I feel like as like a young Indian American kid, I did feel like a lot of kind of family pressure to do something that would be considered like respectful and sustainable. You know what I mean? And that definitely factored into it. Even though I did have like an organic kind of addiction to the subject matter, I felt like that factored in. But the main thing was that I love music so much, and like I was always on the verge of dropping out from being in graduate school or being a professional scientist at some point, to just play music all the time, play music and record music, because I really really love music. And to this day, there's part of me that's a little bit sad that I feel like I never took a proper leap of faith and just see how it went.
Yeah.
And so I was always kind of ready to jump ship to just to be a full-time musician. And then I think there's some specific things about the graduate experience that I found very frustrating. So I think being a PhD student in the sciences is very often very nebulous. Like, you're being judged, you're being continually judged and evaluated, and their expectations. But then you're in this sort of like structure-free zone. Like, once you're done with your classes and you're actually doing your dissertation research, you're in this like structure-free zone. You're largely driving your own ship. I mean, I think there's a lot of variation, depending on what your relationship is with your PhD advisor. My particular relationship was, I was kind of going solo. I was kind of driving my own bus largely, and so you feel like you're being judged and evaluated, but there are no clear milestones and metrics, and I found that to be very frustrating and depressing at times.
In what way are you judged in that environment?
I don't know. I just know that you are being judged. Like, people are constantly evaluating you to see, to determine whether you're a promising scientist and a promising investment, and whether you're going to, you're actually going to be able to do this on your own in the future. I know that people are constantly evaluating you, but, I think you are being judged in terms of your scientific potential and your intellectual potential, but it's really hard to understand how your day-to-day activities are contributing to that. And I found that very frustrating. The kind of amorphous nature of the science PhD when you're done with your classwork was, on some level, the freedom is awesome, right? Because there isn't the structure. You're sort of in your own sandbox, and you're building your own adventure, but it's a limited time. And if you don't succeed, who knows what's going to happen with you. And the other thing too is that,
Well, I'm sorry to ask you this question, but so I was listening to this podcast with Arvind Srinivas, who's currently the CEO of Perplexity, which is, I don't know if you heard of Perplexity.
No.
Yeah, it's a competitor to Google and ChatGPT, and they do something very different in the way that they present the information. If you search on like a particular question, they give you the citations for everything.
Wow.
That they're kind of getting it from. So I think it's a very scientific process in that way, right? You're getting exactly the source of where you're getting this information from.
Yeah.
And awesome. He did his PhD in computer science, machine learning and all that. And he said that, the academic currency, like in the community, is citations.
Yeah.
It's driven by that. So
yes.
I think I've thoughts about this.
You're coming to my next point, which is that the and I suffer with this to this day, to be honest, is the academic currency to me is often not very satisfying. It doesn't kind of fit my biochemistry and my motivation structure. So I think, publishing a esteemed journal is kind of like, that's like the main currency for us.
That is success. When you say, when you talk about the success, that is success.
That is how I think largely how we're measured is like, how many papers do you have in like, what are considered high impact journals? And what does a high impact journal mean? A high impact journal means that typically your article gets cited a lot more when it appears in these high impact journals, which actually is not entirely true, which is interesting. It turns out that, the impact factor of these journals is driven largely by a small handful of publications, and the rest of the articles that appear in them may not actually get cited tremendously. It's really driven kind of almost by outliers largely, but that's another story. But that currency of like, you're evaluated by kind of how many publications you have in these esteemed journals, that payoff doesn't mean that much to me. Actually, what means something to me ultimately, like more than any of those publications, what I would love is to have some impact on the clinic. Like my dream would be to do something where I change the clinical landscape for a disease. Like I bring some new and important tool to how a clinical disease is managed that improves the lives of people. And that can be through the development of a new therapeutic approach, clinical management strategy, or it could even be like the development of a new diagnostic approach that, makes it easier or more affordable to measure something that's clinically relevant. But that, when I'm done with this incarnation or whatever, that would be the impact that I would look for. And so a lot of the currency in academics, it doesn't really fit my personal biochemical reward system very well. And I think that's been a challenge for me.
When you saw, when I was researching about you, I saw there's this H index, which I kind of understood now, what it means. Again, it correlates with citations, right?
Yeah.
It correlates with how many people, last time I checked, I think it was you had 20,000 odd citations or something like that. First of all, is that a pretty high number? Is that high? I didn't judge.
I'll be completely honest with you. I'm not sure. I think I'm pretty good, but I know people that blow me out of the water. So I think I'm not bad. I'll give you that. I think I'm middle of the road. I don't think I'm like a slouch, but I know people that would just dwarf that, in terms of their citation numbers. So I think that I've done a reasonable job and continue to do a reasonable job. That I would say that I'm in the middle of the road. That's my I think that's my understanding of my citation.
Yeah, I would assume that the reason that's the incentive structure is so that people can build off of the research that people are kind of on the shoulders of others, right? That's the idea, is that it's going to add to the...
Absolutely. And let me make it very clear. Me doing science in my lab or, with my team and talking to each other about it without that information getting out is useless. It's absolutely useless. It's kind of like the tree falling in the woods thing. There's no net worth. And I actually think that it's since ultimately, most of my research is funded typically federally, like through the NIH, it's not, it would be a disservice to the taxpayer for me not to report my findings. But the problem is that, to me, I think it's really, really important to have the information disseminated to the public in a way that the public can actually embrace what's being done and why it's being done, so they can be kind of actively engaged in the scientific process, which actually I think would be way better for society. And, I think we wouldn't be running into a lot of the problems that we're running into right now if there wasn't this kind of ivory tower and there was more efficient information transfer between, the labs doing the hardcore science and the general public. But to me, really the dissemination of the information to the public is absolutely critical. And a lot of times these publications, they do exactly what you say, which is very important, is they guide the scientific process in terms of, you need to know, it becomes a foundation of the questions going forward, right? One of the problems there actually is that there's very little glory in publishing negative results. And negative results are equally important, if not more important, than positive results in terms of guiding future questions. But when you get a negative result, like if I'm in the lab, right, and my hypothesis is that this new protein X has a major impact on susceptibility to SARS-CoV-2 infection, and then I do a bunch of experiments in the lab, and then I find, even though protein X was a well-reasoned hypothesis, there was a lot of circumstantial evidence that would suggest that this protein protected people from acquiring SARS-CoV-2 infection, but then our well-designed, rigorously performed experiments demonstrated it doesn't do a darn thing. If you don't report that, which frankly most of the time doesn't get reported because journals are just not interested in it, because ultimately they're trying to sell tickets to the show too. And so there's basically a lack of incentive structure to publish these negative findings. That information is missing from the record for other scientists. And so that information is not used to guide the development of future questions. That is also a problem. What I was trying to say, so I 100% agree that publication is critical to steer the activities of other scientists. The parts of that I don't find very satisfying, one is that a lot of that information never makes it to the general public in a meaningful manner. And there's no excuse for that. It really should, because science is super jargon rich. So like if I show you any immunology paper, there's a million acronyms in there. I would guarantee you haven't heard of them.
The reason I was asking about if 20,000 citations is a lot or not, I don't know, is because of that. It's like, So I work for a fund that invests in alternative health protocols. So we do a lot of work with investing in specifically in psychedelic research right now.
Yeah, yeah.
And so it's a similar problem there too, where there's a few people that actually know what potential outcomes could come out of this, but the public at large has no idea of what to do in this scenario. So it's a question that I'm selfishly asking: how can you kind of go about disseminating? And I think a lot of it has to do with the fact that it has jargon, and the average person has no idea what to make of this information.
Right. So what you need is you need a lot of people that are heavily invested in science communications where they can process this complex information and then disseminate it to the public at large, which is actually, your podcast is called True Calling. I kind of think that's my true calling, but to be honest, it's not been something that I've really invested in very heavily because I'm pretty consumed with actually doing science. But I
Yeah.
actually think that that's one of the most important things that a human can do is take all of this scientific knowledge that's being generated and disseminate it to the public. One of the reasons why I think that there's a bottleneck there is that again, there's not really much of an incentive structure for somebody to get involved in that. It's actually very difficult to make money in science communications. There's not too many opportunities there. So, who are the people that we think of when we think of impactful science communication people? So, somebody I'd think of right off the top of my head would be like Sanjay Gupta, right? He does an amazing job of distilling, kind of complex scientific wisdoms, through the airwaves, right? Yes, absolutely. Yeah. And then there's, a handful of writers that I think of, like specific writers, even for Science Magazine, that, write really great articles that are focused at a more general audience. And they're magazines, like Discover magazine. I used to love reading Discover when I was a kid. Awesome. Discover popular science. But at the end of the day, there's not enough of a universe of science communicators that are doing this. There's just not really an economic environment that encourages people to engage that. Um,
I would listen to, like, so like, obviously, like, the scope and of my understanding of HIV and any of that has literally been some classes that I took in biology in like high school, and since then it's just a joke sort of thing that like we just like talk about HIV but not really understand anything about it, and we just like there's like I have no idea where to even begin when it comes to understanding HIV. For example, how can I prevent it? I know some idea. I mean, obviously, like ask someone if you're that they are positive or not, obviously, but then also like not no syringes and stuff like that. But I don't really know when it comes to this, like what are the preventative measures that I should be taking on a daily basis or whenever. Like I have no idea, and I'm the average.
yeah. So I mean, let's just talk about that. Straight up, I can define that very quickly. So you said the most important thing: you never share needles. So actually, it's amazing. Like clean needle programs are like just incredible lifesavers. Sharing a dirty needle is a very effective method to share a bloodborne pathogen, right? A very effective method. So when they've had clean needle programs for drug users, they have really dramatic effects. And actually, this is a perfect example of where I think, there's this unfortunate tempest that occurs, when scientific evidence collides with moral and religious beliefs. And, so there's been a lot of pushback for clean needle programs because people think, well, drugs are bad. People shouldn't be using them anyways, but that's not reality on the ground. There are people who are using them. And so do whatever you can to protect them from contracting these illnesses. Right? So first of all, there's multiple routes of transmission. So one, there's sexual transmission, right, of HIV, right? Then there's what you just mentioned. So with the sharing needles, that's called parenteral transmission, right? When you actually share needles, right? So that's another way. And the other way that you could get it, but it's a very, very, very rare. So you may remember or have seen, in the history books that when the AIDS epidemic, first came on the scene in the early 80s, there were several instances where people sadly acquired HIV infection through blood transfusion. So through receiving a contaminated unit of blood. And so that's a very efficient transfer of pathogens. Things like HIV, hepatitis B virus, hepatitis C virus, those things can get very, very readily transmitted through contaminated blood, right? So, kind of go backwards, but look, so the one way that we can protect people. So for instance, hemophiliacs, right? Hemophiliacs have to get blood transfusions very often. So if you go back 40 years, you'd see that hemophiliacs, had a very high rate of HIV acquisition by unfortunately being exposed to contaminated blood products, right?
What's a hemophiliac, by the way?
So hemophiliac is somebody that does not produce. Actually, is it factor VIII? They don't produce. Actually, want to get this right, what the right factor is, but they don't produce basically they're missing a clotting factor. I think it's called factor VIII, but they're missing a clotting factor. So if they get
Oh,
cut,
this is Princess Diana. This is Princess Diana.
yes, exactly. That's
Hemophiliac.
exactly right.
All
Yeah.
right.
I think it's called
Okay,
factor VIII.
okay.
Anyways, they're missing a specific factor in their blood so that they don't clot appropriately. And so like if they get in a car accident or something else, they can just bleed out very quickly. And so because of that, they have to get, blood transfusions from people, where they do have that factor that allows their blood to clot appropriately. But since these people had to receive frequent blood transfusions in the 80s, there were many cases where they were contracting HIV infection through this. So one immediate way that you can avoid getting HIV infection is to protect the blood supply from being essentially from all of these transfusion transmitted viruses, right? I actually so I have this, dual appointment. I work for both UCSF, and then I also work for Vitalant, which is a nationwide blood banking company, very similar to the Red Cross. And so I work for what's called Vitalant Research Institute, which is nested within this nationwide blood banking company. And what our institute's mission is to protect the blood supply. And one of the main things we do is we focus on pathogens, like emerging pathogens that can be transfusion transmitted. And we develop novel strategies to detect them ultra sensitively to make sure that we can eliminate any contaminated blood. And we also develop strategies to eradicate those viral pathogens so they're no longer a concern for transfusion transmission, right?
Yeah.
So one thing you can do, and HIV is the history of HIV is a beautiful case of this, where there were very sensitive diagnostic tests that were created. So you could sense, very, very small amounts of HIV in blood. And so what you could do is you just run these tests on the blood when it's obtained, and you can quickly determine whether there's a contamination or not. And if there's any HIV in there, it goes in the garbage, and then you make sure that it doesn't enter the blood supply, right? And so that's done for several pathogens, including hepatitis B virus and hepatitis C virus. So every time if you go to Vitalant or the Red Cross and donate blood, and actually the interesting question is, when there's a new emerging pathogen, is that going to be spread through the blood supply? So this was a huge question with COVID-19, right? So when COVID hit, one of the huge questions was, is this going to be spread through the blood supply? And so my institute, and I didn't do any of this work, but my close colleagues at my institute, they screened like millions of blood samples because we have samples all over the country, right? But mainly we think of it as a respiratory pathogen, right? It infects the airways. The truth is it can actually infect many different tissues in the body, which is one of the reasons why we may have long COVID symptoms, right? Because it does a lot more than just infect your airways. It turns out that some percentage of people, somewhere around 10% of people during acute COVID infection, do become what we call viremic. So they have evidence of virus in their blood, which suggests that it may be transfusion transmissible. And so through this very rigorous, massive study, they were able to unequivocally demonstrate that SARS-CoV-2 is not a transfusion transmission risk, which is why when you go to donate a blood, which everybody should donate blood, that's like a very important thing to do. Your blood
Why is that, by the way?
Why is it? Because there's a huge shortage of blood pretty much all the time. And so what that means is when somebody gets into an accident or they have, some emergency operation where they need blood, there's just not enough blood to go around. And that's a huge problem. So one of the, I think all of us are looking for little things that we can do to contribute. And donating blood is one of the most important things and easy things that we can do as humans to help other humans. Because there's way more of a need for blood than we have blood. That's the truth. And so again, when if you donate blood, your blood will not be tested for SARS-CoV-2 because we've determined that it's not a reasonable transfusion transmission risk, which is a very important thing to demonstrate because theoretically you could say, well, why don't we test the blood for everything, for every pathogen we know, because there's a huge cost, right? And so essentially it would tank blood banking. If we measured every single pathogen, even if it wasn't relevant to transfusion transmission, it would bankrupt the whole system. So one way we can protect ourselves from HIV is by making sure that it's not in the blood supply. Okay, so that's one thing. And then also making sure that people have access to clean needles and they don't share needles. In terms of sexual transmission, one of the most effective things you can do is actually wear a condom. So protected sex almost eliminates the transmission risk. Another thing, antiretroviral therapy. So, if you have HIV infection, we do have amazing antiretroviral drugs. When you take those drugs, although the virus is not completely eliminated from your body, which is why I do what I do. I work on HIV cures because the drugs are not curative. Even though the drugs do not cure infection, essentially they take the virus down to this mathematical level where the probability of transmission is zero by sexual transmission. So have you ever seen, I've seen it like in New York subways and this kind of stuff. Have you seen this ad campaign, U equals U?
No.
So U equals U stands for undetectable equals untransmissible. Okay. And so what that means is that on these antiretroviral drugs and your viral load is undetectable using a conventional clinical assay, it's considered totally fine for you to have unprotected sex with your partner because you're not considered to be a transmission risk at that point. So that's another great thing you can do. You know, it's another great thing we can do to prevent HIV transmission. It turns out that HIV on its own is actually pretty poorly transmissible. If we look at heterosexual transmissions, for instance, like if we look at male to female transmission, and I don't know these numbers perfectly off the top of my head, but HIV infection on its own, let's say like male to female transmission, for any given sexual episode, there might be a 1 in 100 risk if the infected partner infected partner spreading the virus to the uninfected partner, somewhere around 1 in 100. But that probability goes up astronomically when you have other sexually transmitted diseases. So the HIV epidemic piggybacks on other STDs. The reason why it's really maintained is because there's so many other STDs that cause inflammatory like syndromes in genital tissues that make HIV much more transmissible. So one of the best things that we can do as a society to block HIV transmission is actually dealing appropriately with other STDs. Which is an interesting thing. So those are kind of the main prevention measures. You know, wear protection, take antiretroviral therapy consistently if you're lucky enough to have access to those drugs, which a lot of people in resource limited settings who are living off $1 a day or something, it's not yet possible to access those drugs. But if you can access those drugs, taking the drugs is a great thing.
Awesome. This is super valuable. So this is I think is like this will be something that everyone can benefit from. So I wanted to ask you, since you're so into the virus but vaccines as well, about COVID for a second. Because I think it was just a hugely contested situation. You probably know more than I do, and being in the midst of it.
It's been a rough few years. Like conversations it's been insane few years. Yes.
So what was unique about COVID for you? Like the way that it spread? Yeah.
Yeah. Okay. So let me just give you the kind of how things shook out for me. So
actually wait for a second. I see like there's someone waiting for this room. I have another room, but I wanna jump back into this so that we can.
Okay, sure.
Okay, so to jump back into COVID, what was unique about this whole situation?
Yeah, and just the whole experience. Yeah. So first of all, the way that I got into working on COVID. So I think the shelter in place was in March. When everything got shut down, and so just like everybody else, like my lab got shut down, and I was at home, and my kids were at home, and it's like, what do we do now? And then basically what happened was virology labs were thought of as being important work. There's a specific phrase I'm thinking of, but I can't remember. But virology labs were allowed to reopen and specifically conduct work on COVID, on SARS-CoV-2, the virus that causes COVID, right? And so a couple of weeks into the shelter in place, my lab and our institute reopened, and we were working on different aspects of COVID. And so something I'll make very clear: I've mainly worked on retroviruses, HIV, some other related retroviruses, and then what are called endogenous retroviruses, which I'll just mention very briefly because they're so cool, and I want people to chew on it and think about it. But what endogenous retroviruses are? So I mentioned that a lot of how our immune system is shaped is based on these previous encounters with oodles and oodles of other pathogens, including lots of other retroviruses. And I also mentioned that HIV integrates itself into your genetic backbone, right?
Yeah.
As now, there's no evidence that HIV has ever entered your germ cells. Ultimately, when something integrates in your germ cells, then it gets passed on, right, ad infinitum. There's no evidence that HIV has integrated into the germline. But several other retroviruses throughout human evolutionary history, like tons and tons of them, have been integrated into the germ cells. And so if you look at the human genome, the massive human genome, somewhere between 8 to 12% of the human genome is actually fossilized ancient retroviruses that look structurally very similar to HIV, but they're no longer capable of generating infectious particles. But these viruses have literally become part of us. And a lot of it is thought to be sort of like junk DNA. People like to think of it as junk. But it's becoming clear that at least some of these fossilized ancient retroviruses have become part of our own functioning. Like they actually are playing a regulatory role in how our immune system works, which is pretty freaking wild, right? So I had no experience working on coronaviruses before. However, I was lucky to have some close colleagues at my institute that were professional coronavirus people. So they had worked on aspects of coronavirus molecular biology and also coronavirus, what we call serology, which is the study of antibodies that respond to coronaviruses, which is super cool. So it was easy for me to kind of jump in and partner with them and benefit from their knowledge base to start working on the virus myself. And so what we were working on was kind of like how aspects of viral immunology, like how SARS-CoV-2 interacted with the immune system, understanding like what human immune factors were playing a role in exacerbating disease versus ones that might be playing a role in controlling. And then ultimately what we did was we formed partnerships with like a few local pharmaceutical companies to help develop some novel therapeutics for COVID-19. But the one thing I'll say right off the bat, it was one of the most exhilarating things. I've been working on HIV for a pretty long time at this point. So it was super cool and inspirational to be like thrown into this pot, where I had to learn all about the biology of a whole new family of critters. In terms of real life, it's kind of an administrative nightmare in a lot of ways because now all of a sudden you have to try to maintain my HIV research effort and then this other thing. So which means that you have to apply for new grants on totally different topics and develop new relationships and all that stuff. So on an administrative, practical level, it was extremely challenging. On an intellectual level, it was incredibly fascinating, and it was also super cool to just see all of my colleagues kind of nucleate and coalesce around this new public health emergency. And I'm guessing you're probably aware of this, but a lot of what happened in the in the COVID-19 clinical therapeutic and vaccine space evolved directly from the HIV research infrastructure that had been developed for decades. Everything in terms of the therapeutic design, the development of the setup of clinical trials at various sites around the country to even test therapeutics and to test vaccines. So much of that stuff leveraged the HIV research infrastructure that had been built. And so it was very cool to witness all of this.
Wow, I didn't know that. I didn't know that at all. So to kind of wrap this off, I wanted to get a little bit of an idea because I think COVID was a good example of public dissemination at a very rapid rate, and that led to its own set of, I feel like, chaos in terms of you instantly had division in terms of what people think is right for them and their bodies, what to put in there or not. What are your go ahead? What are your
Yeah,
thoughts on that?
this would require 1,000 hours, but basically what you see is because of modern technology and social media, everybody lives in their echo chambers, and then anybody can claim to be a professional. And then you have lots of people with political agendas. Like, for instance, it's very obvious why a lot of people wouldn't want the world to be shut down and businesses to be shut down. It's not a stretch of the imagination, right? And it's also obvious that, why a lot of people wouldn't want to wear masks. I hate wearing masks. I really hate wearing masks. But there's a time and place to do it. And so I think you just have this really awful kind of recipe, where you have people that have access to microphones and social media platforms that really have no respect for science, that have various political and social agendas. And it's very easy for them to find like-minded followers who just reject science. And I think it's the first time we've seen this conflict between science and media on this insane level. But it's extremely dangerous. We can't allow public health policy to be dominated by demagogues that just go with their gut or are making public policy decisions based on religious beliefs. It's absolutely inappropriate. It's really inappropriate. And to be clear, what we saw with COVID-19, this is like we're scratching the surface. So this whole idea of a new pathogen entering the human population and creating a pandemic, almost certainly we're going to see this with increasing frequency over time, and it's because the human population is expanding and we're encroaching on the habitat of lots of different animals more and more and more, like bats, bats, because of the way that their immune system has developed, viruses that can make a living in bats often can spill over into humans. And so the more that humans and bats are living in close proximity, the more we'll have spillover. The more we have livestock in extreme high concentrations, where we have birds and we have cows, that kind of stuff, and humans all working in very close quarters, you're going to have more and more spread of viruses in the human population. So this idea of an emerging pandemic, this is going to happen more and more. And I'm worried that based on what happened here, the next time, like for instance, I'm sure you've been hearing about bird flu, right? The possibility that if the bird flu mutates in a certain way that it starts spreading very efficiently from person to person, which is entirely possible. And some people would even argue that it's not a matter of if, but it's a matter of when. I'm worried that rather than learning from this really traumatic experience that we had, over the last few years, that actually I think it's pushed us backward because people I think are going to be even more resistant to what the scientists have to say and the messages from the public health experts, and that's really very unfortunate. I don't know what the exact solution for the problem is, but it's a very real problem. But public health policies need to be driven by rigorous science. That's the truth. They can't be driven by people's beliefs that they just conjured up. Yeah, I don't know what the exact solution for that problem is. And actually, what we see, for instance, and it's the same thing with this whole vaccine autism nonsense, right? There was one paper that was published, I think, in the Lancet or the British Medical Journal, decades ago, and the author of that paper retracted the article because they recognized that there was a major statistical artifact with the way they did things, and there was no connection between A and B. There was not a connection between vaccine exposure and autism. But for whatever reason, you have these people that will keep putting that paper or that observation on a billboard, and they'll use this to say that vaccines are bad. If we stop vaccinating our children, for instance, measles, it's going to be an absolute nightmare for humanity if we stop doing that. Vaccines have been one of the greatest technological innovations of our time. Are they always going to be perfectly inert where nobody will have any negative effects? That's impossible. You can't develop an intervention without having any negative effect. So if I walk down the street right now and I gave aspirin to 1,000 people, somebody's is going to react to it, right? You cannot have a perfectly inert medical intervention. There's no such thing. But if you look at the COVID-19 vaccine, right, like the Moderna and Pfizer vaccines, and you look at any realistic measures of how many lives they saved versus any quote unquote side effects, from a public health standpoint, it's an absolute no-brainer that they were absolutely the right thing to do. Were essentially a miracle. It was a miracle that those vaccines were developed on that timeline and they were made available to the public within a year of the pandemic showing up.
Thank you for sharing that. Also, last question for you. Do you feel right now that science is losing some level of credibility with the public?
So that's a
This
tough
evidence?
question. First of all, how do we gauge that? A lot of that we gauge through the media too, like this concept that the public is losing faith in science. And so how much of that is, are we getting a disproportional representation of that? I don't know. It's very hard to know what things are being represented proportionally. It sure does feel that way. It does feel, as a scientist, it feels like people are becoming more and more resistant to science. And I think they're resistant to the idea of scientists or science controlling their lives and their daily decision making. To me, a great example of that is the abuse that Dr. Tony Fauci has received, which is just such a shame. This is somebody who's dedicated their entire life to public health, and he's been the target of everything, including death threats to him and his family. So it does seem like there is a lot of resistance out there right now. And again, is it that 50% of America is resistant, or is it that there's a very, very loud 5% of America? I think it's very hard to really size that up based on what's out there in the media. But there does seem to be a very vocal group of people that are fixated against science right now. And I think a very important target for a scientist is to figure out how that communication can be resolved. So there's an increased trust. And part of that, I think, is what I mentioned like a half hour ago about scientists need to make a better effort at communicating their research to the general public, not just sort of discussing our findings at conferences with other professional scientists in our field. We really need to figure out how to distill our stories to the general public in a way that they can appreciate what we do and the rigor with which these studies are conducted, and to understand that, scientists are not evil or not working behind an agenda. But the scientific method in itself has tremendous value, right? So there is not a blind faith. But we don't believe that protein X causes disease Y. You have to systematically prove these things using very objective tools and a principle. And if they could understand that, I think they'd have more trust in science and scientists. But I can tell you this, resolving that is the most critical thing we need to do before the next pandemic hits. for sure we will have multiple pandemics that will come at us. So that trust needs to be in place. And the other thing is we need investments. We actually need investments into public health infrastructure and molecular surveillance so that when the next superbug hits, that we're already kind of pre-mobilized to start, measuring the epidemiologic spread of the virus in our community and understand how it's evolving so that we can actually maintain a repository of therapeutics that are effective at any given moment. That is going to be a tremendous challenge, I think, in the years ahead. It scares me.
Thank you for sharing the time here. I really appreciate this because, I mean, these are kind of conversations that, from an average person that's trying to learn can lead to other people also knowing, and the ripple effects that you're saying that we need more of, like to disseminate the research that's actually out there. And I'm really happy that you brought up Sanjay Gupta and then, Huberman,
Yep.
because they are trying to go out of their way to get this out. And I hope that keeps on happening at a larger scale.
I hope so too. I hope, and a lot of it again is money. Is that there needs to be opportunities for people who are passionately interested in communicating science. From the professional scientists to the general public, those people need to be compensated for their time. So I hope that there's some sort of economic environment that allows for those people to prosper, so that they can get involved in it, because it's absolutely critical to the long-term survival of humanity, the long-term survival and prosperity of humanity. I always think about viruses, I'm an infectious disease person, but as you know, this is equally important in the whole climate change world, right? I think it would have a dramatic impact on how happy our grandchildren are going to be someday.
Thank you for joining. I really appreciate your time.
I appreciate it so much. It was really awesome talking to you. And then I'm sure I'll talk to you again.
Definitely. Thank you for listening.